Thank you for the great introduction. Hello, everyone. I'm Chengxiang Qiu from the J. Shentui Lab at the University of Washington. Thank you so much for giving me the opportunity to present our recent project, a single-cell time-lapse of mouse prenatal development from gastrula to birth. As we know, the mouse embryogenesis is a very complex and amazing process. Just within 3 weeks, a single cell zygote gave rise to hundreds of living cells with different functions and morphologies. There are many interesting questions involved in this process. If we zoom in, focus on single cell at a very specific time point, we want to learn what it is and what's the origin and the cell fate in the future time point. Those interesting questions could be summarized to 2 major parts. First, how do cells transit from early to late stages? And the second question, what are molecular factors, especially the transcription factor, regulating each of the differentiation process? So the whole process of mouse embryogenesis is like a movie, and actually I hope one day in the future we could develop such a technology to put a camera on the embryo to trace the whole cell lineage during this process. But right now it's still not very straightforward. People usually apply the cell lineage tracing or the live imaging for this purpose. However, applying those technology are still challenging to study the whole mouse embryogenesis picture, especially for the much later time points, due to the embryo size very big and the cell population very complex. Alternatively, in the last 5 to 10 years, several studies, they have applied single-cell technology to capture the transcriptional profiles for individual cells in the whole mouse embryo from different time points, including pre-gastrulation and gastrulation, also the early organogenesis. So based on those single-cell sequencing data, those studies identify the cell states for each single time point, and then can connect those cell states across the different time points based on the transcriptional similarity to computationally reconstruct the cellular trajectory for the whole mouse embryogenesis. So this is a very powerful approach. However, those reconstruction results relies on some very important factors. So first, you could imagine the total number of cells profiled, also including the cell number for each single time point. And the second is the depth of profiling, how many cells and how many UMI in the cell. And the third is the temporary resolution. You can imagine sampling every 1 day compared to every 6 hours, totally different. You will miss something during the intermediate cell stage. And also the span of the development is either like 2 days, 4 days, or even the whole time points, 2 weeks. So several studies have been studying the cellular lineage and the trajectory relying on those transcriptome of individual cells for the C. elegans, zebrafish, and the xenopus. However, due to the long developmental time and the larger number of cells in the whole mouse embryo, it's still challenging to study the mice using this approach. So to this end, we thought to apply the similar approach to integrate the 4 published single-cell RNA-seq datasets to collectively span different stages of mouse embryogenesis to reconstruct the whole lineage or trajectory during the mouse development. But when I start to do this work, there is a potential issue that the 2 major data sets, the gastrulation and the early organogenesis, those 2 data sets, they capture cells from the different stages. If we want to connect them, there is some technical batch effect because they are using different technology to generate the data. The gastrulation data, they use 10x and on the cell, but the early organogenesis, which is generated by our lab using the skRNA-seq on the nuclear. To solve those problems, we generate a new data set profile cell from E8 to E8.5. So this time window is the late gastrulation, which is overlapped to the later stage of the gastrulation data set, but we use the same technology as an organogenesis data set. You could imagine such a data set can be used to as a bridge to connect those 2 data sets. And for this newly generated data set, the mouse embryo was collected by Ian Welsh from Jackson Lab, and the single-cell RNA experiment were performed by Ben Smarding from our lab. So this newly generated data allow us to bridge the gap between the two major data sets, and then we could define the cell state at each of the 19 successive stages from E3.5 to 13.5, and followed, we could connect those cell states with their pseudo ancestor and the pseudo descendants. So the project, you can imagine the project is very straightforward. We hypothesize that the distance of cells in the low dimension embedding space, such as the UMAP learned from their transcriptional profiles, can be used to relate the cell states to one another across the developmental time. So after that, we could leverage the linear map we generated here to identify the K transcription factor responsible for every distinct cellular trajectory. So first, we collect the data from the 4 published data sets and first separate the cells by their initial time points, like 3.5, 4.5. And for each single time point, we perform the data processing and the cell type annotation using the marker gene. And next, we connect every 2 adjacent time points, project those cells into the same embedding space. For example, here we project the cell from E625 to E6.5 to the same embedding space. And then we apply a k-nearest neighbor-based approach to connect the cell states between the 2 time points. So here is an example how we create a trajectory from epiblast and its derivatives between E6.25 and E7. So at the first time, co-embedding cell from E6.5 and E6.5, we create a link between the primitive streak to the epiblast from earlier time point. At the second time co-embedding, we create another link between the new cell type nascent mesoderm from the early time point with the primitive streak. And at the third time of co-embedding, we create another link between the new cell type at E7 with their pseudo ancestor. Because we have 19 time points, that means we have 18 pairs of adjacent time points. So we just repeat this approach for each of the 18 pairs of adjacent time points. The resulting graph, which captures the trajectory of mouse embryogenesis, we call it TOM, from E3.5 to 13.5. So in this plot, we showed over 400 distinct cell states and their connecting edges with ancestor and descendants. So in this map, each row is a single cell type and each column is a time point. So before we go in next, I want to say the graph TOM that doesn't reflect the real cell lineage, but just rather the relationship between the cell states which were inferred based on the transcriptional similarity. So despite being constructed through purely computational approach, the resulting representation is largely consistent with our current knowledge of mouse development. You can see the trajectory are clearly separated by different germ layers. From the top to bottom, they are neuroectoderm, surface ectoderm, endoderm, and mesoderm, and actual embryo tissues. So after that, we want to leverage such a linear map to nominate a transcription factor which correspond to a new cell type emergence. So transcription factor play a very important role in determining the specific cell trajectory. So how we do that? So we just split all the map to each individual branch point. For each of those differentiation branch point across TOM, the KTF candidates corresponding to a new cell type emergence were defined by 2 criteria. First, this KTF should increase in the new cell type compared to the pseudo ancestor. And the second, the KTF also should be specific to the new cell type compared to any of the sister cell state in this branch point. So in the subway plot on the right side, I show the top KTF candidates for some major trajectories from TOM. And from this plot, we could learn for those major trajectories for any cell state transition, what are the K molecular factors driving those transition process. So if we zoom in the subway plot, took one example here from epiblast, giving rise to cardiomyocytes through the primitive streak, mesoderm, and the heart field and cardiomyocyte. Every time our cell state transition, it requires a different type of TF involved. So at the same time, we could also visualize the continuous expression level for any individual genes over the cellular trajectory. Here, I show the top KTF candidates to the cardiomyocyte. We could see how their expression level changes at each time of cell state transitions. So finally, to better understand the evolution to shed light on the deeply conserved regulators, we focus on the single cell data set, not just the mouse, also on the other species, zebrafish and the frog. And this is a timeline of embryogenesis of those three different species. We want to find the correlated or aligned cell types and the dominating transcription factor between the different species. So we first just apply the same approach to reconstruct the cellular danger for the zebrafish and the frog based on the published data set. And next, we compare those three danger map or trajectory map to find the correlated cell type between the different species. So here we took two methods. First, just based on the regression analysis of the transcription profiles for each individual cell type. The second way is based on the how many or how frequent of the KTF are overlapped between the cell types. So in the plot on the right side, this is a result. We show the union of a candidate cell type homologous identified by those two methods. Each node here refers to a cell type colored by the different species, either from mouse or fish or frog. You can say they are grouped by different germinals or particular developmental stages. So for example, in this specific bubble, we show the cell type homologous between the three species from the neuron trajectory. Like the sensory neuron from mouse are correlated with the Rohenbeer neuron from a zebrafish and the xenopers. We could further identify some overlapping KTF transcription factor between the cell types that are correlated, such as here as a show example is the neuron progenitor cells. The red color TF are those are shared by the different species. So in this previous work, our goal here is to try to create a continuous roadmap of molecular states in the mouse development. And we systematically nominate the specific TF as key regulators on the initial emergency of each cell type. And finally, we identify the cell type homologous based on the shared regulator or the similar transcriptional states. You can find more detail from our previous paper in 2022. Okay, let's move to the next project. A big limitation of the previous study is that we terminate our reconstructed trajectory at E13.5 because we only have the data stopped at there. At E13.5, there are still many interesting questions that we want to learn, such as when and where each tissue or organ forms its major cell types and the functions. I took one example here, for example, like at the E13.5, we call that a retina neuron and a renal epithelium. And they are terminate state in the tom graph, but we still don't know how they give rise to the so many different types of in the eye and the kidney. We want to learn the trajectory here. So if we go back to see the data, what we have right now, if we compare to major data set, the one focusing on the mouse gastrulation, with cells sampling every 6 hours, but the one focusing on the early organogenesis, they sample the cells with every 1 day. The temporal resolution for the organogenesis data set is much lower compared to the earlier one. And also there is a huge data gap between the gastrulation or early somatogenesis to the birth. So the second data set focusing on early organogenesis was previously generated by our lab, Junyue Cao, profiling 2 million cells from 5 time points between the 9.5 to 13.5. But the temporal resolution, if we want to reconstruct the trajectory, it looks not so good. To improve the single cell data quality, our lab has optimized the old protocol of single cell sequencing, and we test and generate some new data focusing on the beginning of somatogenesis around the E8 to E8.5. So to generate such data, we first collect 12 mouse embryo at this very specific time point, E8 to E8.5. Each of them have measured with a single somite count from a somite 0 and to somite 12. Then we profile there with single cell RNA sequencing to look at their transcriptome of individual cells. So here in the low dimension embedding, UMAP cells are roughly separated by different germ cells where they derive from. Of note, this data has been used in the previous project to pressure the two major data sets. So benchmarking in our lab optimize all the single cell or sky-RNA-seq protocol allow us to profile the embryo with much higher data quality. So if we look at those older data and the new data, they with the similar sequencing depths, but the median UMI per cell increased from several hundred to several thousand, which is very promising. So from this data, we could identify very detailed cell population. For example, the difference of anterior and posterior floor plates. The anterior, you can see the connect to the forebrain and the midbrain, and the posterior connect to the spinal cord. And the different segmentation of the hindbrain, which refers to the Romere 2, 4, 3, and 5. And also the first and the second heart fields, and the three different populations of neural crust, which are derived from different regions of the neural ectoderm, forebrain, hindbrain, and the spinal cord. So the testing data with high quality encourage us to extend the current time series from a somite 0 to 12 to even much later time points to generate a high temporal resolution data. So then we were able to connect those separate stages to study the whole picture of mouse organogenesis. So we hypothesize that the data with higher temporal resolution allow us to reconstruct a more smooth and continuous lineage of cell state transitions. So to generate this data, our collaborator Ian Welsh from Jackson Lab collected and staged mouse embryos with somites ranging from 0 to 34. So each of those mouse has been measured with somite counts, followed by manually checking the other morphological features. After that, after E10, we collect the embryo with every 6-hour temporal resolution until to P0. Those embryos were first staged by measuring their digital size, followed by also manually checking other morphological features. And after that, for each single time point, we select 1 or 2 embryos from those time points to perform the 3-level sky-RNA-seq technology to recapture the single-cell transcriptional profile. So the sky-RNA-seq is a kind of a pull split. By 3 rounds of adding barcodes, we could profile over 1 million cells in a single experiment. Benchmarking from our lab optimized our protocol and perform all the 15 single-cell RNA sequencing experiments with 21 NOVA-seq rounds. After processing the whole data and removing the low-quality cells and the doublets, in total, we recovered over 11 million cells from 43 time points from E8, the early or the late gastrulation, to P0. The data quality is really good with optimized protocol. The median UMI per cell is over 2,700, and the median number of genes detected per cell is over 1,500. And this is the cell number for each of the 43 time points with 6-hour temporal resolution. You could see most of them are over 200,000. And this is the cell number for a subset from E8 to E10. Each of the mice has been measured with somite count from 0 to 34. Next, we perform the dimension reduction and cell type annotation based on the marker gene. At the result in the global view, we identified a total of 26 major developmental trajectories. So from the global insight, the biggest cell population correspond to the brain and the neuron development, like the cluster 1, 2, 5. Also, we identified a different type of mesoderm cells and also different type of blood and immune cells. The same UMAP on the top left, but cells are colored by their initial time points. So for each of individual major trajectory, we further perform the sub-clustering, resulting in a total of 190 sub-cluster with cell type with distinct annotations. So here I want to use two examples to highlight the quality of our data and how we do the cell type annotation. So the example here, we focus on the neural ectoderm and its derived cell types, including like a neuron and other like glia cells. If we took the subset of a cell from neural ectoderm, like the basement cells, and into a 2D UMAP and color cell by their annotations, we could see the cell from a different region of neural ectoderm in the UMAP are roughly arranged based on their spatial locations. For example, from anterior to posterior, they are the telencephalon, diencephalon, mesencephalon, the hindbrain, spinal cord. And on the other axis, from those when the ventral, they are the roof plates and the floor plates. On the other hand, if we look at the thing as neuron cells, the derivative of the neural ectoderm, we do the sub-clustering. We also find a different type of neuron cell. Also, we could, if we took the two largest cell type in this embedding, the excitatory neuron and the inhibitory neuron, and put them to the sub-clustering, we could even find a more detailed cell population. For example, the spinal interneuron from dorsal to ventral, they format to the multiple layers. Those neurons could be specified by the top highly expressed transcriptional factors. The second example from the lateral plate mesoderm and the intermediate mesoderm. So in this view, I show all the potential subtype of from these two types of mesoderm. However, it's really challenging to annotate this cell type purely based on the marker gene, because those are like a different type of mesenchymal and smooth muscle cell, their transcriptional profile are very similar. Also, the study on those cell type are very limited. To address this challenging, we perform the spatial mapping based on the published spatial transcriptome data to predict their spatial location of those subcell type. The spatial mapping results help us to finally understand what they are. For example, the mesenchymal cell, they are from different organs, like liver or lung or heart. The previous example showed the profiling depth, where we also aim to show how the temporary resolution improve our understanding of mouse embryogenesis. So here we focus on very specific developmental stage, the early somatogenesis, including embryo with somate counts from 0 to 34, which refers to E8 to E10. So, and also we focus on very specific cell population corresponding to the posterior embryo development. Because at this time point, the embryo start to elongate to the posterior, and many interesting and important cell population are involved in this process, like NMP, spinal cord, node cord, mesoderm, and gut cells. So here we select all those cells initial annotated as NMP and as well as other corresponding to the posterior embryo cell types into a 3D UMAP, by which we identify the three major cell cluster. The cluster 1 refers to NMP, as well as the spinal cord progenitors and the mesoderm progenitors. The cluster 2 refers to the node cord, and cluster 3 refers to the gut cells. And the same UMAP on the right with cells colored by their somite counts. You could see that even just in 2 days from E8 to E10, transcriptome of NMP are changing so fast with every somite count increasing. So here we dig a little deeper on the NMP cells. NMP is a very interesting cell population identified during the somatogenesis at the most posterior side of the embryo. The NMP has a bipotent differential cell fate, either giving rise to mesoderm cells or the neural ectoderm cells. So here we perform the subclustering on the cell from the cluster 1 and visualize them in the 2D UMAP with cells colored by their initial annotation on the somite counts. The cells are distributed based on the different transcriptional angles. So first, you could say the left express SOX2, which is a marker gene of the neural ectoderm cell fate, and on the right express TBX, which is a mesoderm cell fate marker genes. On the other axis, you can see in the middle express T, and on the two sides is MES1, which refers to the progenitor cell state or the mature cell state. On the other hand, you can see the bottom express CTX1, the top express HOXA10. The CTX1 is an early somatogenesis stager marker, and the HOXA10 is a later stager. So if we perform a simple PCA on those cells, we could say the top three principal components explain almost 50% of the total variance, and they have very clearly meaning correlated to the specific meaning. For example, the PCA1, you can say is correlated with the TBXA and SOC2 expression, refers to corresponds to the mesoderm and the neuroectoderm cell fate. And PCA2 correlated with the somatic count increasing, refers to either early to late or the anterior to posterior stage. And also the PCA3 correlated with the gene T and MES1 refers to a differentiated state and undifferentiated cell state. So with this data set, we show the temporary resolution power and the cell, for individual time point, how many interesting things we can capture. And we sought to investigate the cellular trajectory during the mouse organogenesis. How do three germinals, ectoderm, mesoderm, and endoderm from a late gastrulation develop into different mature tissue or organ close to the birth? The first example here, we focus on the renal kidney development. There are two major trajectories involved here. The anterior intermediate mesoderm gave rise to a collecting duct system, and the posterior intermediate mesoderm gave rise to other like tubular or glomerular cells. And this is the same UMAP color cells by the time points. And because the UMAP here, all the trajectory are relatively straightforward, so we could manually reconstruct the linear relationship of those cell types. The two linear, they're giving rise to different type of renal cells. The second example is the eye development, those different type of retina neuron and the pigment cell, they are derived from the eye field from very early time point, including some major trajectory like the retina pigment, amacrine horizontal cell, photoreceptor cell, and the retina ganglion cells. And the retina neuron subtype also present a temporary feature. They appear at a very different time points. And we could also manually reconstruct the linear relationship of those cell types based on the UMAP, what we observed here. However, some example are much more complex. For example, for the blood trajectory, here I took all the subset of different type of blood cells and re-embed them in 2D UMAP, all the subtype, including the astroid, T cell, B cell, macrophage, they appear derived from HSC. So compared to the previous renal or eye development, we found the trajectory of blood genesis is much more complex. It's challenging to draw the similar graph manually. We need to think about some systematic way. So here we thought to apply a more systematic way to reconstruct a cell type relationship. So we count the mutual nearest neighbor identified between each pair of two cell type with this subset. For example, we took one example, the trajectory from HSC gave rise to MDSC through the two intermediate cell state. Those color cells are the mutual nearest neighbor cells identified between each two adjacent cell population. The hypothesis is very simple. You could imagine that the cell type, if they have a larger number of mutual nearest neighbor cells between them, they are tended to have a linear relationship between them. So this is a developmental trajectory for the blood genesis, which is generated by the mutual nearest neighbor based method. So we just repeat this process for the different systems like blood genesis, neuron genesis, epithelial cells. Then we could combine them after that and generate a pseudo-tree for the whole picture of mouse embryo cell types. So here we also integrate several published single-cell RNA data from early time points, including the pre-gastrulation and the gastrulation. So the tree finally described the developmental trajectory for the mouse embryogenesis from E0, a single legger, to P0 over 100 different cell types. Actually, if you look at this tree, in each cell type node, the cells, because we merge the cells from different time points, you could imagine it's very heterogeneous. For example, the HSC with CD34 positive is the ancestor node defined connected to the different type of blood cells. In the UMAP, all the cells are from HSC, but they are colored by the mutual nearest neighbor cell if they are progenitor cells to giving rise to different other cell type nodes on the UMAP on the right. And some TF and the gene, you could say even still at the HSC, but they already show highly expressed in those progenitor cells. So this inspires us how to nominate the KTF corresponding to the new cell type emergence. So based on the cell type trajectory, the big map, we sought to systematically nominate the KTF and the K genes, which are potentially regulating every cell state transition. So first, we split every cell state transition from A to B to three stages. The early transition happen in the ancestor node, like the HSC, and the real transition between the two nodes A and B, and the later transition happen in the children node. And then we check which gene increasing or decreasingly expressed at each step of a transition. So this result could be benchmarked by some pseudo-time analysis. For example, this is the trajectory from anterior primitive streak giving rise to the definitive endoderm. And the cells are colored by the pseudo-time. The SOX17 and the CER1, they identified as highly expressed in the early transition from anterior primitive streak. And they are upregulated earlier along the pseudo-time compared to the left genes. In the last section of this paper, I want to share some really interesting finding. So when I analyzed the data, we identified a abrupt transcription changer for cells from embryos before birth, right before birth, and right after birth for some very specific cell type, including the liver, adipocyte, and the lung. You could see here on we show the three column, the first two columns corresponding to all the time points before E13.75 and right at the 13.75. The last column refers to the P0, which means right after birth. You could see the P0 cell from these three cell types, they are far distinct from other cells from the early time points. Next, we perform a k-nearest neighbor-based approach to globally check every cell type, which of them show the similar pattern. Again, the top cell type will show changes sharply. After birth are mainly from these three different tissues. In contrast, multiple types of a neuron, they don't change too much before and after birth. So we vary because our data, especially for the P0, was generated by a different batch of experiment. Potentially there could be some batch effect. So to validate this finding, we perform another new experiment and generate some new data. We collect another non-mice. All those non-mice are from a single mom. 3 of them are naturally birthed. And after that, the left 6 are from a C-section, but profiled at 0 minute, 20 minutes, and 46 and 80 minutes. For those 3 specific cell types, we found a very similar shapes of cells over time after C-section. You could say especially between the C-section 40 minutes and 60 minutes, you can see the transcriptional changes a lot between those time points. So then for those 3 cell types, we also want to learn what kind of gene or molecular mechanism change those after birth. So we identified a differentially expressed gene either between the E13.75 and the P0 in the original big data, also the genes which are correlated with the C-section time point in the new data set. So many genes are overlap between the two approaches. They are correspond to a very specific molecular mechanism. For example, the genes identified in liver are closely related to the gluconeogenesis regulation. In summary, in this work, we generate a comprehensive single-cell dataset for mouse organogenesis and annotate around 200 distinct cell types. So we focus on the developmental trajectory for specific tissue like renal, eye, and also we build a pseudo tree for the whole mouse embryogenesis. And finally, we identify some transcriptional change for specific tissue right after birth. You can check our paper, which is published recently, for more details. For the next plan, recently, Sylvia has published a review paper who was a former postdoc in our lab, introduced a new concept to define the cell types. Traditionally, cell types have been defined based on their morphology, function, or anatomical location. However, in this review, she proposed a new concept that uses a developmental tree to define the cell states based on their lineage. This could including also cell from the aging state or the disease state. And this is a really promising idea, but it requires the data and a well-designed strategy how to reconstruct such a comprehensive tree. So our next step is to extend our current analysis from P0 to A0, and maybe in future could even to D0. So we could capture the whole picture of mouse development process. Finally, I want to thank the people who contribute to this project from Chengxiang Lab and all our collaborators, including Ben Smarting from our lab, perform all the SkyRNA experiment. Ian Welsh from Jackson Lab collected and staged all the embryos. Okay, that's all. I mean, know if any questions. Thank you for your patience.
All right, thank you, CX. That was incredible, incredible work. Really exciting. Yeah. I think if you have questions, please put them into the Q&A. I have plenty of questions. Although, as I was thinking of questions, I was finding it difficult not to gravitate toward my organs of interest. Like, just I've got a million questions about like, hey, how does the otic vesicle get patterned and that sort of thing. One kind of, I think, general interest question is, in development, we often see these kind of converging cell fates arising. And in one interesting corner of the embryo that we study is the development of the cranial neural crest and how we see emergence of these, they're called ectomesenchymal cell types, which resemble a lot of the cell subtypes you see arising from the different mesoderm layers. Have you looked at all at that sort of phenomenon in these data and gotten any deeper insights into that type of process?
Yeah, thank you for the question. I would say, first, we generate the data and publish the data. We are not the people who can understand all the data. We welcome everyone. For example, like some people, they really interested about neurogenesis. Some people, they working on the heart. They can help us. We can work together to dig deeper into this data set. Like we focus on the kidney, so we took some time. Like you said, like cranial neural crest, which also very interesting here. Like you say, the convergence is a really interesting question, like the endoderm convergence to the gut. Also like you said, the neural crest, which is I would say the most complex cell population in the development process. I didn't really dig deeper, but it's potentially we can extract the very particular time point, like early somatogenesis, and extract their closed cell type, like the mesoderm cells, like potential neuron cells, to do the subclustering. Maybe we can find a more detailed structure there. Yeah, but that's future work.
Yeah, no, I guess to that goal of collaboration, maybe you could tell the folks joining today, like, about access, how usually, I haven't checked out this one in particular, this new data set, but is there like an online portal we can go to and just look for our favorite gene? kind of zoom through the data, or are we gonna have to pull this from Geo and analyze it ourselves?
Yes, I can show if you are interested. We have very good.
Oh yeah, that would be great.
Can you see my screen here? So here, which you can choose like which data set you interested. I show the different the major trajectory. You can either visualize by the 3D UMap, or you can also download the subset of the data. You can either like, for now, you can also check any gene count from the UMap. Also, you can from the link download the subset of data. And also I also created those example from, for example, the kidney, eye. Yeah, I think because you say the first, you could say the data really big. We tried our way to split it to the different part. People, if they focus on the kidney, they can download kidney data. Second is because our approach is a whole embryo profiling. So how to split the data is really based on my kind of annotation. Like maybe people can make mistakes. So that's all my work. But if you have found any question, I'm always happy to hear. So we can work together to improve. Yeah.
Yeah.
So Carl, can I ask a follow-up question since CX actually showed the website here? So recently we actually tried to build a web portals for some single cell RNA-seq data and spatial transcriptomics data. We are curious, like the web portal you are showing here. Do you have any like pipeline to do that, or you basically use any published software to create this web portal? Yeah,
so this website is created by one former graduate student for our lab. I think she has created a GitHub to show as a template, and people can easily download that to you. But it might need some manually change or something, but I don't think it's really hard. Yeah.
Great, we'll explore that.
Yeah.
Thank you.
Yeah.
Yeah, that's great. Yeah, having published several of these single cell studies ourselves, I realized looking at your data that like one of the things I was marveling at is just like the organizational challenge that all of these data present, and how well you've been able to kind of parse it out and make it comprehensible to a reader.
Thank you.
And it's fantastic that you've made all of this, even just like developing a site like this, I think is a very valuable resource to the field. So thank you for that.
Thank you so much.
Yeah. I had a question about the resolution of the data set, and kind of, have we achieved the maximum resolution? Like where do we go from here, as far as, do we need to just redo this entire process all over again, and then have like multiple replicates that we can compare to get even deeper insights? Or do you think you've hit the mark as far as the intervals between the different developmental stages to get the maximum amount of information?
No, that's a good question. I think at the beginning of my talk, I say some important factor which affect the performance of a reconstruction. The total number of cells and how deep we sequence the cell, and the temporary resolution. All those are very important factor. Also, current data, as you said, even just based on current data, we just dig deeper for a part of those information. The second, I don't think in my opinion, the temporary resolution doesn't need really, same resolution. For example, at the early somatogenesis, you need a higher temporary resolution. But when you close to the birth, seeing the change are not so fast, potentially you don't need such a temporary resolution. Also, at the early stage of potential, you don't need many cells, but during the birth, the mouse embryo is getting very big, and also the cell number, the cell type population, you need to profile more cells. And as you said, in next step, if people they still want to look deeper in this through, they could very specific time point maybe do deeper sequencing or profile more cells or higher temperature resolution. But the whole process from E8 to P0 maybe not quite necessary to reach, especially I found at a later stage, like it doesn't change really too much. You could focus on your own specific question. Like for me, potential, I will look into the translational changes right after birth. I potentially will generate some new data looking at like C-section, or right after birth, like a P0, P1, P2. We can see some other changes during this process.
Yeah, that's a fascinating research direction to take this in. Oh, it looks like Xiaomi's got a question.
Oh, sure. Yeah, I think this is really comprehensive work and great talk. So I have one question. Like, I think like when you mentioned about when you build the hematopoiesis trajectory, you actually analyze the data from E0 to P0, right? Because like all the other parts, yeah, I'm talking about like E8.5 to P0. So I just wondering about those early time points. Like, did you collect the data like probably I'm not sure if I missed like it's from other people, or like you collect the data similar to the later time points, and if it's different people, like how did you integrate the data with those early time points?
Yeah, so yeah, that's a good question. So here, as I mentioned, for the data from E8 to P0 from our current data set, but for the early pre-gastrulation, the gastrulation from other published data, because our SkyRNA has some technology limitation. We cannot work on the very small embryos. Also because the gastrulation data set, like from the Pigeon Sarah's Nature paper, it's already has a very good quality. And for the before gastrulation, it's even harder to profile those cells. So we just connect those published data sets and do the data integration. But here I didn't mention that we didn't include all the cells to create this tree. We first split to the subsystem, for example, neurogenesis, bloodgenesis, and gastrulation stage, pre-gastrulation stage, and within each subsystem, I created those trees. And then after that, I try to connect those trees to the group to form this big tree. So it's not collecting all the cells do the same thing at once. So that makes, yeah.
Thank you. Yeah.
I had another question about so one of the interesting uses for a data set like this would be that we could think about, especially the information the way you've gone about identifying different transcriptional regulators. We could think about doing in silico perturbation experiments where we see what happens when you knock down or upregulate a gene of interest or transcription factor of interest. A lot of the kind of systems that have been developed around that are integrating both RNA-seq and attack-seq data. Where do we stand as far as our ability to do that using this particular data set? And are you making a similar version of this with attack data?
Yeah. Thank you for the question. Actually, yes, we have the attack data, which I'm recently working on. So it's not ready to publish yet. And at the same time, as you mentioned, the perturbation, we had a paper last year. We profiled the embryo with a knockout versus the wild type. We profile them with single-cell RNA-seq and dig into like a cell type combination changer or gene changer and use experiment to validate. At the same time, we use some in virtual system to profile the larger screen on the TF function. For example, like knock out each single TF to say what kind of linear change. So we have those projects going.
All right, cool. Maybe you can speak to the challenges with the attack data or integrating the attack data. Are you running into?
Yeah. So actually, usually you can imagine if you have those aligned data set, if you have RNA-seq, when you analyze the attack-seq, it will help a lot, right? You could integrate the RNA-seq and attack-seq, which help on you understand the cell type, what they are. You can also compare the TF expression and the TF their their activity, like a binding motif on the same cell type over time. I think it helps to better understand like a gene regulation network or something else.
Yeah. So we actually recently, analyzed some single cell RNA-seq data and the single tax-seq data. I think Chenghui is here also. Chenghui is basically mainly working on that project. We actually use Surat pipeline and also I think I forgot the name, Signac, to do that kind of single cell tax-seq analysis. And then there's one question from the reviewers to suggest to use Cynic. I'm not sure you have used Cynic or not, or any other software you actually familiar with or suggest to us to use. I think Cynic is one of the software actually proposed by one of the reviewers. Seems quite interesting. Have you ever used this software before? It seems like they can integrate in a single cell RNA-seq tax-seq data together and then predict this kind of
I see.
factors.
I haven't tried that before, but as you said, there are so many different you can do this, like integration, either like attack RNA-seq, spatial, and other. So when I do my analysis on my data set, I first try the Surat, but I don't know, maybe because our Sky RNA-seq or Sky attack, the data is sparse compared to the 10x generated data, so it works not so good. I use another software called SCGLUE, SCGLUE, which is developed by, I cannot remember, but that's really, I find the integration works pretty good. So they based on some graph-based deep learning method to integrate the two models data. That works pretty good. Yeah.
Great. Yeah, good to know that.
Nice.
Yeah, in terms of so many data you have, like so many time points here, single cell RNA-seq data, attack-seq data, and feature spatial transcriptomics data, the integration part is kind of quite important.
That's true. Yeah.
Yeah.
Yeah, we're one step closer to a future where we don't actually have to do the experiments. We can just crunch the numbers.
The AI will do everything for us. That would be good.
Yeah. All right. Well, I don't have any other questions. I'm not seeing any questions from our quiet audience. But I really wanted to thank you again for joining us. I think this was fantastic. I will send you a list of questions about the diversification of cells in the otic lineage. But I won't bore everybody else with those questions.
Great.
Okay. Yeah. Thank you for the invitation. Very fun to share. So yeah.
Great. Well, thank you both. Have a great day.
Okay.
Thank you.
So bye.